Hi, everyone. I think we'll get started. Very enthusiastic about this town hall. A little bit different format than how we typically do, which is just kind of open-ended and fielding questions and giving updates. And that's in part because we have a pretty big milestone to celebrate as a community. We've had our first clinical trial data readout for SCN2A, so very exciting. And to kind of celebrate that and kind of raise awareness around the data and what's going on there, we have a few members from that biotech team, Praxis Precision Medicine, on the call to give a quick introduction to themselves and also to kind of go through the initial data from this trial. Praxis Precision Medicine is a biotech company currently with 2 drugs in the clinic for SCN2A, Prax-222, which Marcia will be talking about today, and Prax-562, a oral small molecule that's also in development for SCN8A and is still enrolling patients. So Marcio Souza is the CEO of Praxis Precision Medicine. I'll hand it over to you.
Thank you. Thanks, Sean. Thanks, everyone, for being available on a Sunday. Very appreciative of being here and having a conversation with all of you. So I'm going to share a couple of slides. I think today's conversation is less about slides and more about your questions and what you want to talk about. So I'm going to try to go through that fairly quickly. And I hope you can all see it in the proper presentation mode. And I'm just going to move you on my other screen here so I can see your faces a little bit better, if that's okay. So what we are working towards here at Praxis, right? Praxis is a biotech company, meaning we are small in size, science-driven, really energized by getting drugs to patients. And it's very personal for most of us, myself included. So it's something that we really care every single day about keep doing for this community. I wanted to start with a couple concepts, maybe before we move into some of the updates for 222 and where we are with 562 and 222 in general, right? Which is pretty obvious and might be pretty clear for all of you. But I always like to remind everyone, what is the conditions for successful drug development and science and the unmet need for patients. So the right science, the commitments to do the right, and having unmet needs to fulfill is on the core of all of that. But there are thousands, if not tens of thousands of disease that meets that criteria and never actually went anywhere. The second part is the funding and making sure that the conditions, like the people and everything else that is necessary for drug development, is in place. And I think we've been pretty good on making sure that both of those things were in place and continue to be in place, right? The science should always lead, but we want to make sure that we have resources to get these drugs to the kids and young adults and hopefully late adults that are necessary. And obviously the mobilization from the ones who actually need this the most or want this the most, that they are the patients. And incredibly enough, I think that's where sometimes it doesn't work. I think that is a little bit of discussed truth about this interface. And it's part of that I wanted to field questions today as well, because I think it's super important to understand what are the needs, how can we work together towards these needs, because at the end of the day, there's only one thing that matters here, that is the patients getting drugs, right, that are actually positive for their lives. When you think about 2A, and there's an N missing on the title there, so my apologies for that. It's been pretty clear, the commitment from Praxis, right? The 2 drugs in the clinic, as Sean mentioned at the beginning, 562 on the right-hand side here, functionally selective drug for focus primarily on reduction of seizures, but normalization of hyperexcitability as the goal. And on the left-hand side, we're going to be discussing this more today as 222, what I would say The difference between what we'll have on the top, that is 222 is genetically targeting early onset, and 100s that is targeting loss of function or autism phenotypically for these patients, is just a stage that they are in development. But as you can see, like there is not even another single program in the clinic for any other institution. I would say, unfortunately, right, the more developments, the more research on these fields, the better. But for a company like Praxis to actually have three should speak volumes to everyone. It definitely speaks for us on the commitments for this. So if you go back to the previous slides, getting all those elements together is fundamental. One program success enables the next program success. One program learning enables the next program learning as well. And I think that's why this ecosystem is so interesting. If I go very quickly about the basis and why we're excited about two of these programs, right? You might have seen this before, but this is like a very quick, some preclinical work on 562. Once we had on the left-hand side the very clear activity on a SCN2A model, right? So it's not a generic model, but like one defined, showing incredible efficacy, may I say, preclinically. On that, there was tons of things that had to be done in the middle, like preclinical safety, preclinical pharmacology, clinical safety in 2 phase 1 studies in adults, all the formulation work and so on, to lead to what we are right now, that is a pediatric friendly formulation that really no one else has on any of the drugs being developed in this state, being in the clinic. And by being in the clinic, what we mean is going through all the hurdles, right, to be able to have, from a safety perspective, from delivery perspective, from an ethical perspective, to be able to dose the drug to patients, especially vulnerable population, not only by being pediatric, but by having a DE. And what is the newest part of this equation was a lot of feedback we got about, like, it is very difficult, and you can reinforce for us it's correct or deny in this conversation for parents to actually get these kids out of the home and bring them to a faraway site and so on. So we create an entire infrastructure to be able to execute this study at home. Like, we're lucky enough that with 562, by the weights that is delivered, as Sean mentioned at the beginning, it was possible, right? And this is going on. I want to make sure to reinforce that we see sometimes these molecules, as I mentioned on the previous slide, that separate, but they are really not, right? Like all the learnings in each one of them are able to generate and to leverage for everything else that's being done. Didn't go for background, but my experience being pretty much all my life in drug development for smaller indications. So I've seen firsthand one molecule enabling many other molecules, many other programs, like, and incrementally, and then hopefully at the end, really working towards normalization for these patients and these kids and adults on where we are today with many of these other diseases. Unfortunately, not yet for SCN2A DE. The data for 222 preclinically is equally compelling, right? As you look here, it's like on the left-hand side, this is all animal data, so obviously preclinical in vitro in cells and so on is obviously very positive. But what you see is very impressive reduction in seizures, just like the previous slide with 562. And what I would call a time-independent rescue of the phenotype, which is incredibly important, right? Because what you see in many other drugs for other disease is that if you give at day 1 of the animal life, it rescue, but if you give at day 10, it doesn't. And we know that, like, everyone in this community and in other communities out there, like, you don't have the luxury to just have like newborns at the time of being treated, or a month-old at the time being treated, but different age. So by establishing early on that you could be able to rescue, if it all pans out in the clinic at different ages, is quite important. And the whole grail, I would say, of any genetically defined therapy is that you normalize, right? So that you get very close to what we call a wild type. And that's what you're seeing on the bottom here. I know this is fairly busy and was not meant to explore each one of them. What I was trying to say is there is a path towards things that we all want for as kids, but all those paths, in our view, they go through reduction in seizures, because that is really the way the brain's telling us, like very loudly, that something is going on. And by going there, by reducing the seizures over time, we would be able to arguably reduce other like symptoms or normalize other symptoms that we would be looking for. And that is obviously a big deal because we want to make sure that we go towards certain goals. So where are we with 222? It's the one, as Sean mentioned before as well. We have like some data, right? We started the program like many years ago, but it got to the clinic like earlier this year. And the idea was twofold. So first is to establish that by giving to patients intrathecally, so in their spine, a dose every month that was safe. And then that we would decide or not that it had to change the frequency that the drug is given or the amount that the drug is given, right? The dose or the regimen, or both, or not change at all. When you translate, and by translation I mean when you go from like an animal to a preclinical safety in a non-human primate or like monkeys to people, there are a lot of assumptions we use. Like the size of a juvenile brain, for example, is about 10-fold smaller than of a human. So you make that scaling when you're doing, but those animals are normal, right? They don't have a mutation like leading to like hyperexcitability or something else in their brain. So there's a lot of calculations that are done there. So as good scientists, we always assume we are wrong instead of assuming we're right. It's a cautious thing to do. So we have a hypothesis, but we always assume that there's some margin there for actually not being correct. And we want to be as safe as possible while creating as much benefits as possible. You might have seen this on the previous presentations we gave that as life goes, as I always say, we're planning everything to go one way, but it ended up going a slightly different way because we got an emergency access request from an unusual case, a kid that was diagnosed intrauterus, so before birth, with a 2A DE, for other reasons actually that have very little to do with what we know. So we got the request. This baby was on status at time of birth, and just to give like a general idea, right, that the medical team tried everything that was possible. As you all know, that's normally what happens. Very good medical team, but that was the very first patient. It's very unusual to get the very first patient to be an emergency case versus the clinical trial case. The good news, as you might have seen this before, it's a very risky proposition for a company to actually allow emergency access as the first use, as you can imagine. And we can discuss that more on the Q&A parts. But the first dose, which was about a month old, the baby one was dosed. They was well tolerated. That was the number one thing that we cared about, right? Make sure safety first. And we start seeing some of the episodes of status, which was a pretty consistent and continuous status to be subsiding. But then after 7 doses, that what you have on the right hand side here, it is continued to be pretty safe. And there was a number of other gains on SCID, including like a complete cessation of status. And while I'm using 50% reduction here, I think it's actually not fair to say that way because that was way more than 50%. It's just because it's impossible to calculate when it's ceasing completely to not ceasing. What is the actual percent, right? When you divide something by 0, it's obviously not possible. And then a number of other gains that happen. Now, this is all great, and obviously every kid or every person who can help will feel very happy, but we need to go back to the cause of really helping a community and a number of patients, not only one. So when you go back to this study, that's how it was defined. 4 to 6 patients at the beginning, they had to have functionally defined gain of function, not only genetically, but like biophysically. And those of Praxis 2 at 1 milligram about every month. And when I say about is 28 days, could be like a day more, a day less, about every month. 4 doses were predefined. And remind what I mentioned a couple of minutes ago, the goal was to establish that it was safe, right? It's always the first goal when you're doing something like this. Obviously, that's not the ultimate goal we all want. We want to be safe and to help people, but that was the first goal here, was to be safe. What you've seen after 3 doses, the 4th dose is being wrapped up, the data right now, you're soon going to be able to see it, was something that it's unprecedented on any drug developments for these in general, or for many other genetic diseases, that after the very first dose, and I'm going to jump back to the previous case, which after the first dose was benefit as well, we're seeing a very significant benefit on the hallmark of the disease, right? 100% of early onset patients have seizures. There is nothing that get even close to that, and that's why this is, on our view, on the regulators' view, quite important endpoint, and it's been consistent ever since. The other part that was quite important for us, because we heard from many of the parents, like you all, that having days without it was important. More days without it allows for like better care, better family dynamics, and so on and so forth. So there was a very significant increase on the number of days without any seizure on these patients as well. Now, there's a number of other things that happen that we've been collected, and we're going to be presenting pretty soon, once they are all compiled, in terms of what the impression from the families were on what was happening with these kids as they're taking these drugs, which for us was incredibly important as well, because it's not only what I call the tangibles, right? It's the things that we can measure and that the regulators care, like the FDA, but equally important, what's happening at home when like all these kids are having interactions with their families as well. So we're going to be talking much more about that in the near future. Now, none of this would matter if the drug was not safe, because like you can have like certain results, but if it's not safe, there is no way to move this forward, right? And that's a lot of the drugs actually die on this stage when you can actually achieve something, but you cannot do that safely, right? So the concept that regulators use is benefit risk. So it's always a ratio between the benefits that a drug gives and the risk the drug gives. Now, there's a number of acronyms here, right? That we can unpack on the Q&A as well, but basically, when we are in this stage of development, any stage of development, what we are trying to do to understand is whether or not, when you give the drug, if it elicits a side effect, if it starts a side effect, right? So we'll call drug like related side effect, or treatments emerging adverse events. So after you give the treatments, and it's after, meaning after the patient's in a trial. It does not mean the day after, the hour after. It means if they are receiving a drug, did it happen? And then we have to ask ourselves, is that associated with the drug? Meaning, we all get like flu, and some of us got COVID and things like that. Those are obviously not associated with the drug. But there are other things that could be, right? Certain toxicities or inflammatory markets and things like that. So we haven't had any of those events that are associated with the drug right now. which is quite remarkable. We don't need not to have, if I can use almost a double negative there, like it's not a requirement not to have, like actually all drugs that are effective out there have something that is listed by the drug, but it was very good to see. And we have a safety monitoring board, or it's a DMC, a data monitoring committee, that are independent physicians that look into this data and say whether or not the drug should continue to be given to patients, because obviously we're biased. We're biased to want, even if we want to do this in a most scientifically rigorous way, we do have an interest for this drug to move forward. So it's important to have an external view to always tell us, no, you are correct or you are incorrect. And so far, this view is being basically the same. So what we take from here, and that's basically my last slide, so we can jump into your questions, is completing all the data collection, which is imminent. We need to go back to the FDA, because one of the things is our previous study design for the next phase is irrelevant right now. And that's the thing about science as well. Like, you sometimes learn on the negative side, meaning it's less than you expected. And sometimes, in the more rare case, like this one, it's doing a lot more than you expected, a lot faster. And there are ways to have a discussion with the FDA to accelerate that and to get that faster to patients, which should be our collective ultimate goal. So that's what we're doing. And the idea is to start a pivotal trial next year. Just logistically, that's the fastest it can be done. Like, I would start today if I could, but that's the idea. Now, I'll go back to the very beginning of the conversation, right? When you talk about like what is needed, the science clearly is panning out. I think this is probably the worst possible moments in biotech history for funding, but we're going to continue to figure out, because the science is there. And part of this conversation is, if this is important for patients, not only for 222, but 562, but even for loss of function, right? This is one win, I'm going to call this way, scientific win and positive win from clinical, from patients, a win for the entire community, even outside of SCN2A, but precisely into SCN2A right now. Like, how can we make sure that we are all even more mobilized together to actually get these treatments there, in our view, but I'd love to hear yours. These kids definitely need right now. So I'll stop right there and field any questions. And I hope this was productive.
Thank you so much, Marcio.
Of course.
Really appreciate that. A few elements I just wanted to kind of clarify for the audience. With biotech companies, like the funding element, like the funding isn't asked for funding from the community, it's from investors.
Right.
And how that typically works is, investors, as they get more excited about a drug and molecule, are willing to invest more into biotech companies to continue to develop it. So I just wanted to clarify that one element. One other element too, just from messages, this is preclinical data, meaning that you can't get it at your pharmacy yet. It's still being tested and evaluated.
Right.
And I definitely agree with Marcio. Definitely a major win for science with their initial data. And the hope is that this drug will get developed and then approved by the regulators for each country, and then it will become available at your different pharmacies. Or however else you'll get it, which I think this will be probably through a hospital. And we did get some questions through that. Marcio, if you have the chat open, I'll just kind of start with the top and walk through these for comment.
Yeah, I just opened the chat here as well, so let me know if you want me to go one by one, or if you prefer to do that, Sean.
Sure. Yeah, I mean, you can go one by one. I think the first one is by Jenny Burke with the FDA.
Yeah. So Jenny, maybe I'll separate this into hypotheticals and clear ones, right? Every single drug for epilepsy in general. So if you take the DEs as a subset of epileptic conditions have been approved on seizure reduction to date in the US. There's an established path on what they would like to see, how they would like to see, and quite importantly, fits very well with the data we know from natural history from this community as well. While the ultimate goal of treatment is not only to reduce seizures, should be one must ask, and for us was very important to ask this at the beginning, if we should look into something else for approval, right? And I think that the answer for us quite definitively was no. The relative prevalence of any other symptom would never make to the top. And what I mean by that is, while we know the constellation of things that happens with these patients, there is not a consistency that would allow for actually good understanding what it means to, if one patient reduce a given symptom and another one reduce another symptom, like collectively, if we're measuring A or B, it did not like actually get anyone better, which is, it's an unfortunate like way, but it's still pretty much collective statistics that get like the drugs to be approved in the United States. I do believe that many other things are going to continue to be explored, but I also believe that they might take a different amount of time to get there. And then when you have two variables, that is, one is we don't know exactly what to measure and we don't know when to measure, the answer should be quite definitively, do not do it, right? It's a disservice for the community to do that versus service to that. So that's why we ranked pretty high for us. And I think for others developing drugs in DEs as well to seizure on this case as endpoints. Now, since I said it's all connected, I'll finish by saying for the loss of function, it probably cannot be, right? So we need to learn right now, as we're going to be going towards like a loss of function development, hopefully soon, on what needs to be measured, but going through the same scientific rigor of what is the most prevalent, what is expected to change based on the mechanism of the drug, and what is the timing that is reasonable. Illustrating that is, the drugs that have been approved for other epilepsies in the US were approved with 3 months data, right, 12 weeks. And it's pretty much what I just showed to you, is being sufficient with larger number of patients, but being sufficient for approval. Many of these other endpoints take years to change. So then you have to ask the questions like, do we want to stay in development with potentially placebo, with for 3 or 4 years observing, when there's a question mark at the end if it was even valuable to measure, versus something that is certain, that can allow for better collection of the data over time. Sorry for the long answer there for the question. So, Stefania, is that the next one, Sean?
Sure. Yeah, Stefania for the next one would be great.
Yeah, so any improvements in developments, capabilities like walking, talking, interacting. So I'll start with a more personal, and then I'm going to go back to the drugs. I find it's like, I would love nothing more than what we see is normalization, right? It's like, if you like go back, and I not even like using the word normal, because normal associates to something that that is so like unclear to all of us, but I would love to see improvements in all these areas. And when you look into the translational data, they works on those other endpoints, right? Like ultimately survival, but like normalization of what we call phenotypical manifestations in general. I think it's early to see, not necessarily early to measure. And what I mean by that is we're going through in all the case report forms, right? What the trials collect, looking for other improvements as well. And that was what I was trying to say before. There is much more that we need to talk about in the near future. Ultimately, for both 562 and 222, and hopefully 100 soon, would expect that by normalizing the like hyperexcitability, we allow the brain to actually continue to develop in other areas. We're not there yet. I think what we have right now is the reductions in seizures and increase on seizure-free days. But there is nothing telling us that we shouldn't get into other improvements in the future.
Thank you, Marcy.
Of course.
I think the next question is from Carla Forbes.
Carla, yeah. Would all of the drugs be available for patients who are not having seizures? Yeah, Carla, I would say yes, right? And that goes back to when we're actually able to move these developments to a potential approval, and then we can start to explore even more things that are necessary here. It's there's an old adage I always use. Sean probably heard me saying this 100 times: is the best drugs are the drugs that make it, all right? Because then you can continue to understand and to develop, and allows for second generation, third generation, other mechanisms too. So eventually, and I'm very happy that there are a number of patients that are not presenting with seizures anymore. But we know the costs of that, right? And the cost of that is like oftentimes like heavy sedation and inability to do like other things, because we're really dumping the activity of the brain so much on that regard. And none of the drugs that are used right now as anti-seizure medications are specific for NaV1.2, for example. So what they end up doing is interfering with a number of other things as well. So making the first transition that is like more specific drugs, and then the second transition that is like, okay, now even if you don't seize, can we actually get a general like reduction without interfering of the hyperactivity, without interfering with other functions, is the ultimate goal. And I think we're going to be there eventually once this drug progresses in development, but it becomes a necessary condition for that to progress, for us to get to that point. I hope I answered your question, Carla. Okay, I think there's Michaela and Aaron here. When will the next injection be given for patients who are currently on the 222 study, right? So four injections were what the FDA allowed us to do initially. We are working as hard as you can imagine to actually get back to them with the package to be able to continue to give these injections. I think it will be irresponsible for me to tell you a date. What I can tell you is we will be in front of them pretty quickly, and I fully expect that they're going to see what we are seeing, like the benefits here in the preclinical package. We're going to be able to go back with a date pretty soon on when the next doses are going to be able to be given for patients, which for the ones who were on the trial, right, the idea was to quote unquote move, and I don't mean move physically, but just like theoretically to what is called cohort C of the trial. There's a long-term dosing as well, and we're looking forward to be on that phase pretty soon. Okay, hope I answered that one as well. Let me find the next one. Anita
Morris.
Would the drug work for those with later development seizures? So at this point in time, Anita, the most important part is whether or not is a gain of function mutation. So very, very rarely you have a more clear manifestation after 3 months. The vast majority of these patients can be diagnosed by just having a seizure before 3 months of age, as you all know. But if there is a doubt on that, we would be more than happy to facilitate through the channels, for Sean and others on. If the mutation is not characterized biophysically, we would do the biophysical characterization and would get back to you. And if that's the case, then yes, then it could be used. But it's quite important that it's gain of function in the channel. There is a lot of people who I would say with good intentions attempt to characterize those mutations. I would say it's very complex to characterize. So I would urge you to actually pay attention to that, because it's going to be important for all these genetically defined ones. The next one is from Michael and Aaron as well. What should the patients and parents do? I will wait for the next dose. So I wish I had a better answer for you, but I really do. But I think one is make sure that you continue to document like what's happening, the kids, with your kids specifically, if that's where the question is coming from. Continue to communicate with the sites. I believe public advocacy matters as well, and more than happy to discuss that. And I'm sure you can discuss that with Sean and Leah and the rest of the foundation here in terms of what can be done. I do think there's been very little public advocacy for 2A. Like if you go to the Hill, for example, there is no one who ever even heard that this is a problem, that drugs have to be developed, that the FDA should pay attention to it. So there are individual things can be done, like constant contacts with the sites, which in the case in the US is in Memphis, like Dr. Willis' site, but then there are more things collectively as a community that I believe could be done as well in terms of general advocacy and raising the awareness through the FDA and otherwise on the importance of the research to continue.
Thanks, Marcio.
Can I just, Marcio, I'm sorry. Can I just also just jump in there? Sorry. So Marcio answered the question from both an advocacy standpoint and the current families and patients that are in the 222 study. But if there are additional families that are interested in 222, we do have the website, which you guys saw on the slides, embravestudy.org. It's still live and active. And there is a nurse there that you can speak to directly about your individual case. And you can still go there and kind of be lined up for the next stage, and we can contact you when that opens up. So I do want to encourage, that's a place that families can go right now if you're interested, and we'll keep you updated.
Yeah, thanks, Ross, for that. So the next one, either drug works for then with, we tackled that one, sorry for that. Could there be additional benefits with fully controlled seizures? So it's interesting, right? It's all a little bit hypothesis, what we were saying here, but in the study, in the cohort we had, I think we had the most extreme case, the one I mentioned on the emergency access, where it was like no subsiding of the seizures. And then we had patients that were, I would say, reasonably well controlled. And I mean reasonably because well controlled would be no seizures ever, like grade developments and so on. And they were like obviously in other medications, right? So one would imagine there was like not a lot of gains to be having on that. And it's not the case. It's actually, we're saying like good gains. We are not there yet for 562 because the trial is blinded. We're actually still very much looking for people who are interested, especially in the US, to participate on that study. So like put a plea out there that it's quite important to answer that very question to get like people interested. But the idea is that what's happening right now is like we're using a hammer with a pin needle. That's what is happening with the current medications, right? It's like you're blocking every current through the channel. And of course, sometimes it works because you're like sometimes you can get that to go where you want. But what you're attempting to do is actually to thread like something through the needle with 562 and 222. So they're being a lot more precise. So the question is, because we got those questions before, why are we being so strict on the inclusion criteria? Why are being like requiring like so much control in terms of the dose, in terms of the number of ASMs? Because it all goes back to the comment I made a couple minutes ago. Like if I come here 6 months from now and tell you there's barely any benefit with 562, I don't think you're going to be too excited about that. And even if I explain to you at that point in time, well, there's no benefit because there is no currents on that brain. Like we dumped so much that there is nothing there. So what we want to create is a condition where they can get better, right? And that is being the balance that they're trying to achieve here is treating to a point that a drug can actually help. We all seen this, right, for our lives, for our kids' lives, is like that is that next drug that actually makes things worse. And then there's an entire rebalancing of the system that has to be done. And we're working very hard for these drugs to do much better. And in certain cases, it's going to be by combining with other drugs. In most cases, it's going to be by removing certain drugs that are actually not being beneficial for them on the grand scheme of things. So that is the, again, that is the long answer once again for Oliver for that question.
I often like to think of this one as when you have like a knocking in your car, but you only notice it when you go above 65. You know, you can drive below 65 and not hear the knocking, but you're not going to really get to where you need to be very often and miss a lot of meetings. What you really need to do is take the car in and get the transmission fixed or check your calipers. And that's the kind of concept for these next generation drugs is you're not going to just slow the car down to not hear the knocking. You're going to go in and fix the actual part.
That's exactly right. That's exactly right. Yeah. So I appreciate Leah putting the study links there as well in the chat for ones haven't seen. I'll ask you to go there and to share around people you know, and I will be more than happy to, even with the simple description of the mutation. I think there was a question about whether or not, like a given for Zainab, whether or not would be gain or loss of function, even just with the description, right? Don't need to know where it came from. We can make the constructs, we can test, and we can tell you if it's in our database already, can tell you right away. If it's not, we'll do that there is no cost or there is nothing involved with that. We're creating kind of a general database to actually inform development in general for this community. Yeah, so I think the next question was the shelf life, and Michaela and Aaron, let me just make sure I understand your question correctly. So I'm going to give you the pharmacology answer to that because I believe that's what you're asking, right? So there are a couple parameters that we define a drug. One of them is the half-life. It's like the time to achieve like half of the concentration after you give the concentration to a patient. And there are 2 or 3 dynamics here with 222 that is slightly different from other drugs, right? So, for example, if you give phenytoin, which might not be a great example because it's erratic, the distribution, but you know the concentration increase after dose, it starts to decrease in general, and it's a matter of hours, right? That's why for the most case, you give like 3 times a day, and you're trying to keep the concentration above a given threshold. There are other drugs that in the beginning, that's what happens. So I'm going to use like cenobamates that some of you might know, right? It goes up and down a little bit. You start very, very low. After about 12 weeks, you reach therapeutic levels for that drug, and we call that like steady state, therapeutic steady state, and it stays pretty much there, but takes a very long time, but you get to concentrations. With 562, for example, to continue on the small molecules before we jump to the ASO, it stays for much longer, higher once you give a couple doses, and it accumulates. And what I mean by that is, after you give certain dose, it's increased a little bit more than just the dose you gave. So you have a little reserve, if I may, on staying there. And the goal on 562, for example, is not to have a lot of variability, because what we know happens is seizures, breakthrough seizures in general, they happen when it goes too high or too low on the therapeutic concentration. So we're trying to keep that very tight And that's one of the major benefits, actually, that we expect from 562. None of those drugs that I just mentioned, they stay for very long in the tissue. So that's number one. And the other one is that there is no secondary effects, meaning they bind to the channel, they unbind, and once the concentration goes down, that it's not there anymore. Why I went through all of this? Because 222 is different than that. When you give the drug, it starts a process, right? A genetic process. So the actual half-life of the drug, which is about 30, 40 days, as we expected, the dose that are being given, is now followed by the process half-life. So you have the drug, and then you have, obviously, at the end of the day, what we want here is to have less channels that are like active. Let's put this way to simplify on this one. And that can last for a few more weeks as well. So you have the drug concentration itself, but then the mRNA half-life that is different, protein half-life that is different, and they all follow each other. So it is, you do all of those things mostly in monkeys, right? Because I'm sure we all would agree that it's a really bad idea to get a piece of tissue from a human, especially when you're talking about the brain. So they're imperfect. That's what I'm trying to say. Like, all these translations are imperfect, but we have a window between like about a month to about 2 months for what we know today that the drug would have effects. Now, the longer you go beyond that, the lower the general concentration is going to be. Right? So what we are seeing right now is after one dose, we see a very significant reduction in seizures. So we know we're already there from a active perspective. So we want to try to keep as much as possible aligned to that. Like, we don't know what's going to happen if you go much lower than that. And can still be reasonably positive, but we definitely don't know what happens on the other side and if it still would be positive. And that's why when we find a positive signal, you try to understand as much as possible about that over time by repeating that dose, by observing more. Because since there is a lag, as I just explained, you want to make sure we are taking advantage of that lag as well as drug developers. And I know this is all very frustrating, right? Because what he wants to do is, once everything gets resolved, and now we can just go and develop this drug, gets that to patients, but it takes certain steps to really understand exactly what's going on. And I think we are there, like in a much, much faster way than it's actually quite incredible. Like, if you were to ask me 2 years ago, would you be here today talking about meeting with the FDA for a pivotal trial? And I'll tell you, very unlikely. And so we are on that period. So now we want to make sure we answer the questions we still need to answer. That is probably larger numbers, how they behave versus different regimens or different doses or things like that for the time being, at least. And Kela and Aaron, let me know if I answered the question about how long should stay by going through that long explanation. Is there a yes? Yes, okay.
One other element that it's just an observation from my standpoint. I think kind of the initial thought is that we'd have to knock down or reduce the number of SCN2A channels by more. The starting dose that Praxis did, as Marcio elegantly explained in the beginning, was small, to be safety cautious. But the magnitude of seizure reduction with that small dose was meaningful. And to me, what that means is that maybe smaller changes in SCN2A can have a bigger clinical effect. But translating that to your question, Michaela and Aaron, that opens up the door, at least in my mind, that with the half-life, that you may have a longer therapeutic benefit from the molecule, with that degradation over time. I think that would be my hope.
Yeah, I think that's ours as well, Sean. And where are the moments that we know, because I do feel, maybe I'll detour for a second here. There's been a push for longer, right, in drug developments in general. And I think with channel disease, I would say, if I could characterize that way, that principle is not clear yet. So I actually think it would be more dangerous to go much longer than it is right now. At this point in time, it might be that I'm completely wrong. I said at the beginning that is, we normally start by thinking we're wrong, and then and you go and you test it. Because we have no idea what happens on the other sides of this, right? We know, because nature run the experiments, that patients with just a slightly change on the gain of function, right? But we know that they are phenotypically outgrow their seizures. They are like phenotypically much better. So what we are seeing to some extent with 222 right now, and we're expecting to see with 562 soon, is that we really didn't need a lot of the drug in the system. And one like dream scenario is that you can just solve the phenotypical like difference from wild type over time by giving a dose that is fairly safe versus triggering on the other ends like something that is unwanted. So I think that that's the balance. Like, I hate using the word balance, to be honest, because we're not attempting for balancing here, right? You have the kids that are like in needs of getting as much resolution or full resolution as possible. So that's not balance. That is really driving towards maximum efficacy. But what I mean by balance is making sure it continues to be safe. And that's where we care about driving as much efficacy and continue to be safe.
Makes sense. All right. Are there any more questions from the audience? And we did record this, so we will have it up and available for the community if anyone wants to rewatch. And if anyone has any questions subsequently, we're always here. And if we don't know it, we know the people at Praxis to ask to get their opinions and their answers.
All right.
All right. Well, thank you, Marcio, and thank you, Ross, for all the effort into 2A. It's definitely a celebratory, point within the SCN2A community of first clinical data, and fingers crossed for continued success and that we're able to get drugs for our children soon.
Absolutely. That's why I wake up every day. So we're gonna be there soon.
Thank you, guys.
Thank you. Have a wonderful.
Thank you, Sean.
Take care.
Have a
Bye
good
bye.
weekend.